Here.
Cool.
Okay, we are live. All right, hi everybody, welcome back. This would be part 2 of building a protein. So depending on if you're at home, this is what you'll be watching for the activity. So we have our amino acids that we put together based on the quality of our group from last time. And so what we're going to be working with is our purple tuber that we are going to place a blue end cap on one end and a red end cap on the other. So the blue end cap represents the N terminus, which is the beginning of the protein, and the red end cap represents the C terminus, or the end. All right, so we got our beginning of our protein. Just don't forget, so we're going to start. And then our end. All right, so what we want to do now is pick a random assortment of 15 amino acids. So we need 6 hydrophobic, 2 acidic, 2 basic, 2 cysteines, and then 3 hydrophilic. Okay, all right, so I'm going to move this out of the way. Now we can do that. All right, so mix them together. And then we're going to place them in any order on our tuber. So we want to put them about 3 inches apart. And once we place them down, we are not to move them. I feel like this might be the part that's hard, though, because then they're supposed to identify which ones we stuck where.
Or should I?
No, it's all, I mean, it is what it is. I feel like that's where they'll just have to pause. So, any random order. Or we can also, I can download this and send that to them too.
Okay, yeah.
So that you have a.
That's a good idea.
Yeah. I think I might turn this into an Ed puzzle too.
You're not going to fit.
Make it work, Mr. D. I feel like you might have to go back to the top. I was like, well, no, because did you start 3 inches in? Yeah, so like just
put one at the end.
Yeah, like there you go, boom.
Done. Okay.
Okay, so now what they're supposed to do is record the amino acid sequence of the protein, starting at the blue end cap. So I kind of feel like we need to bring this a little closer, and maybe you guys will have to just pause as you try to identify them.
What each one is.
Yeah, so number 1, 2, 3. Try to turn these so you can see them better. 4. 5. Caught on something. 13, 14, and 15. All right, so again, you're going to use, do you want to pull that up? Amino acid chart to determine the order of the amino acids. So again, you might have to go back and rewind and pause to get through that. Okay, and so in the lab, you should be answering the question, what level of structure did we just create? So, like guess it is what it is. Secondary structure would be a little to represent by the folding into alpha helixes or beta sheets, which we could do in this lab, but it's a little bit more than we're trying to accomplish right now with what we're demonstrating. So secondary structure would be alpha helixes or beta sheets, but we're going to be skipping this part. All right, so what we're going to be doing now is folding our 15 amino acid protein according to the chemical properties of its side chains. And remember that all of these qualities are affecting the amino acid at the same time. So we're going to start with folding the hydrophobic side chains. So remember, our hydrophobic ones have the little yellow tabs to them. And hydrophobic means that they want to be buried inside the protein. They don't want to be exposed to the water molecules on the outside. So we're going to fold it to make them hidden or be inside of the protein.
I guess we're just doing that for right now.
Yeah, that works. Yeah. All right. All right. So next thing would be the acids and bases. So the acids and bases are charged side chains. So what we want to do is place the negative side chain with a positive side chain so that they become within 1 inch of one another. This is turning out to be too simple. All right, and keep note that as we continue to fold our protein and apply new properties, some of the side chains that we previously positioned might not longer be in an ideal place. So we have to move to compensate for that. All right, so the next thing would be that the cysteines are positioned opposite one another. So I feel like we're just going to bend some stuff. Exactly. All right, and lastly, the hydrophilic side chains, which are white, is to make sure that they are exposed to the outer side of the protein where they can bond with water. So I'd say, yeah, or if we want to kind of kink this now.
Oh, right.
Let's kick this out that way.
And then this stays out, but that one has to go in because that was an in. Right?
Yep. And then same thing, like let's do a crazy bend.
Okay.
Like
so if we do like that,
yeah.
And then bring this back around like this. Yeah. Right. So that's on the outside. Right.
And then we still have to have our cysteines together.
Our cysteines still have to be together.
And then this also. So let's kink this in here. And then maybe. Does this work?
Right. Yes.
Okay.
Yes.
Okay. Good. And then we still have our.
Oh, no, we still got this problem now.
Okay.
We got to bring our acids and bases need to be next to each other. So where was this other? Where's the other blue one? Oh, yeah, down here. So they're going to be near each other. They're going to want to be near each other. This guy's going to want to be inside. So.
And then we have our greens still.
Our greens are still together. That's in an outside.
Okay. Look good? All right. All right. So final shape of our protein right here. And the question we're asking you is, what structure is this representing? And that is it. Oh, well, I guess the would be what's quaternary. Actually, we can just add that. I think the easiest